So we have seen how the lipids are digested, and then they are divided into smaller particles, and after the process of emulsification or absorption, they then move towards liver for their final catabolism. Now, the catabolism, the major type of catabolism of lipid, or their major catalysis of lipids, that is known as the beta oxidation. Why we say beta oxidation is because this is the beta carbon of the fatty acids. There is a alpha carbon, beta carbon, gamma carbon, and so on. So the carbon which is at the position number beta, that gets oxidized, from where it gets the name beta oxidation. It also, it's important to know that this pathway takes place in the mitochondria. Why? It shouldn't be difficult for you to know that why I asked why. It should be very easy for you to know. Yes, mitochondria is the major organelle in which the process of energy formation, ATP formation takes place. This beta oxidation produces CoA, NADH2, and FADH2. Acetyl CoA, I mean. So acetyl CoA, you remember, is the 2-carbon moiety, which takes part also which actually start the process of Krebs cycle. So first of all, you should keep in mind the environment in which this is taking place. On one part, you are in the cytoplasm, and you have to move your lipids towards, and which are mainly the long chain fatty acids we are talking about, and you have to move them towards the mitochondria so that they can move into the mitochondrial matrix. So there are definitely certain barriers which the nature have to devise ways to move the lipids from the cytosolic environment to the mitochondrial environment, and the barriers are of course in the mitochondrial membranes. So first of all, what should happen is that if we see that the fatty acid should first be converted into fatty acyl CoA through an enzyme which is known as acyl CoA synthetase. So once it is converted into fatty acyl CoA, then it moves to from the outer mitochondrial membrane. This one is the outer mitochondrial membrane. It moves from the cytoplasm towards the mitochondrial space by moving through the outer mitochondrial membrane. Now, this fatty acyl CoA has to, as I have told you, its final destination is the mitochondrial matrix where it has to move. Now, the thing is that fatty acyl CoA cannot move through the inner mitochondrial membrane because somehow it doesn't have the machinery. It is the inner mitochondrial membrane resists it for this movement. It acts as a barrier. So what will happen that then there is another molecule known as carnitine. So what will happen that the fatty acid part of the fatty acyl CoA will be then combined with the carnitine to make a carnitine fatty acid conjugated molecule, while the CoA is released. In this way, this carnitine helps in the movement of this long chain fatty acid towards the mitochondrial matrix. Once into the mitochondrial matrix, what will happen that this carnitine is released here, and you get the free fatty acid. This free fatty acid is once again then combined with the CoA to form fatty acyl CoA through the activity of a second enzyme known as CPT2. And in this way, because of this shuttling of the carnitine, this fatty acyl CoA is able to move in the mitochondrial matrix. And once in the mitochondrial matrix, it now will be acted upon by different enzyme to carry out the process of catalysis of long chain fatty acids. Also keep in mind that, of course, any chemical pathway we are talking about, it have many regulatory steps. One of the most important regulatory molecule for this carnitine shuttle is the malonyl CoA, which acts as an inhibitor of CPT1. So in this way, this malonyl CoA gives a signal to inhibit what we say, the movement or transportation of the long chain fatty acid. And you can understand why it is so. Why malonyl CoA is acting as an inhibitor? Because malonyl CoA is the compound which is involved in the synthesis of the whole biosynthetic pathway of the fatty acids. So because of this, now it gives the signal that now synthesis is required, the synthesis of the fatty acids taking place. So at that point, degradation is not required. That's why it acts as an inhibitor. The carnitine is an important compound, as we have told you, that it's involved in the transportation of lipids. Because of this, which the degradation of lipid is possible. So it is supplemented in many drugs in case where you need the lipid, where for instance the lipid degradation is not taking place, or it's not correctly taking place. We will talk about the carnitine deficiency later. Then what about the short chain fatty acids? Short chain fatty acids don't require any real mechanism because they can easily be diffused from the mitochondrial membrane, so they don't require the carnitine shuttle. Now, very important, once in the mitochondrial membrane, the long chain fatty acid we undergo the oxidation reaction known as beta oxidation. For instance, here we are taking an example of a 16 carbon compound, fatty acid compound, and what will happen that this is, you see here, keep in mind that there will be 4 reactions which we must say takes place repeatedly in beta oxidation processes. First of all is the oxidation producing FADH2. Oxidation takes place here and you get FADH2. Fine. As a result of this oxidation, so you get in the a double bond between the beta and gamma carbon molecules of the fatty acyl CoA here. The second is the hydration process, that water is added, this hydration process, and as of course the enzyme hydratase is involved in it, and you get hydroxycyl CoA, and because you get an hydroxy group added. And then a second oxidation step takes place, but this time it's not FADH2, it is NADH2. You get NADH2. And finally, there is the splitting of the molecule, that is the thylalytic cleavage, which releases the acetyl CoA. In this way, because of these 4 steps, first is the oxidation by FADH2, second is the hydration process which add water, and third you get second level of oxidation through NADH2, and finally the thylalytic cleavage, which ultimately gives you the acetyl CoA. This process is repeated again and again and again, and with each process you get acetyl CoA, and eventually you end up at the very final step, end up with 2 acetyl CoA molecules. Right? Now you see so much FADH2 is produced and so much NADH2 is being produced. It shows that this degradation of the long chain fatty acid must be giving very much of the energy, and that is what actually happens. If we, rather than seeing this slide, I have take to this one, it will be more easier for you to understand. So here you can see here each cleavage of the thylatic cleavage, which ultimately give rise to the formation of one acetyl-CoA molecule. You, in this whole process, you get FADH2 molecule, NADH2 molecule, and also the acetyl-CoA, which goes into the TCA cycle. So if we are talking about 16 carbon atom molecule, that is 16 carbon atom fatty acyl-CoA, you will have what we say the 7 FADH2 will be produced, fine, and similarly 7 NADH2 will be produced. 7 FADH2, you know, each FADH2 contributes toward 2 ATP molecules, you will get 14 ATP molecules. Each NADH2 molecule gives 3 ATP molecules almost, you get 21 ATP molecules. Each acetyl-CoA which goes into the which goes into the Krebs cycle, it gives you 12 ATP molecules. So because there are 7 7 repeated, this process of 4 steps is repeated 7 times, so you get 96 ATP from the TCA cycle also, and ultimately you end up with 131 ATP molecules from the degradation of 116 carbon fatty acyl-CoA molecule. Keep in mind that because 2 of the ATP molecules are used in the start for the activation of the acetyl-CoA, so you finally end up with 129 ATP molecules. So at the very end, you get 129 ATP molecules. But still, it's a huge amount of energy you get as compared to what you have learned until now. In glycolysis, you got only 2 molecules of ATP. In what we say the Krebs cycle, you got around 34 molecules of ATP, and here you are getting 129 molecules of ATP, which is really many times more, which shows the importance of degradation of lipids or importance of lipids as energy source.